welcome our speaker for today, Emmy Wesdorp. She is postdoctoral bioinformatician and translational researcher based in the Netherlands. She's working at the intersection of molecular diagnostics, infectious diseases, and pediatric oncology. She is affiliated with the Princess Máxima Center and collaborates with the University Medical Center Utrecht, where her research focuses on developing non and minimally invasive liquid biopsy approaches for pediatric oncology. Her most recent PhD was conducted at UMCU under the supervision of Professor Jeroen D. Ridder, that is detection of pathogens through liquid biopsy shotgun sequencing, where she explored how sequencing cell-free DNA from blood and other body fluids can identify bacteria and fungi in challenging clinical scenarios. And this will be the focus of her talk today. With that, welcome, Emmy, again, and please take it away.
Thank you very much for the kind introduction. So yes, as already explained, so I will talk about my PhD thesis, which was focusing on cell-free DNA shotgun sequencing for pathogen detection. And actually today I will focus on short-read cell-free DNA and how we try to find the needle in the haystack. And I will do that by first still giving a little bit of a background, although a lot of it is already told. About my scientific background, then talk about pathogen detection and clinical need for improved diagnostics. I will focus thereby on invasive mold disease in immunocompromised patients, especially in pediatrics, and show some of our proof of principle work, which is focusing on enhancing the capture of fungal cell-free DNA, but also some in silico approaches that we are working on to strengthen pathogen detection. And I will also tell you a little bit about our future plans to showcase what we're planning in 2026. So as I already explained, so I'm indeed a researcher that is based in the Netherlands, specifically in Utrecht, which is located in the center of the country, and I'm affiliated with two different centers. So the UMC Utrecht, where I conducted both my master as well as my PhD, as well as the Princess Máxima Center. And the Princess Máxima Center is the only pediatric oncology center that we have in the Netherlands, which is currently doing most of the care of our cancer patients within the Netherlands. And I'm actually working there now as a postdoctoral researcher, mostly in bioinformatics. So mostly next generation and third generation sequencing. So indeed, my PhD was mostly under supervision of Professor Jeroen de Ridder, where we focused on next generation sequencing and liquid biopsies to detect bacteria and fungal pathogens. What I'm working on now mostly is liquid biopsies to support risk stratification, support targeted therapies, and improve longitudinal monitoring of pediatric cancer patients. So we focus in the current job more on the ctDNA levels within neuroblastoma and other cancer types, such as the renal cancers, which is under supervision of Professor Lavage. And actually also we're collaborating between the two labs to see if we can bring the liquid biopsies to a next level. So as I explained, today's presentation is mostly about detection of pathogens through liquid biopsy shotgun sequencing. And most of the figures are shown also are integrated in this thesis, which I will later give you a QR code to actually have access in case you are interested in reading a little bit more. So as I mentioned, the overarching theme of my work is cell-free DNA. And I thought it's good, just in case that people are not so familiar with what cell-free DNA actually is, to give a little background on this type of molecules that we use for real-time monitoring. So cell-free DNA consists of very short and encapsulated DNA fragments. They can be found in all kinds of body fluids, including urine, cerebrospinal fluid, but also blood. They are released upon different pathological and non-pathological processes and become available upon apoptosis, necrosis, and active secretion. And the interesting thing is with cell-free DNA, it's kind of a minimally invasive tool for monitoring of what's ongoing at that particular moment in a particular patient. A little bit more about the characteristics and also why I think it's a very suitable candidate for real-time monitoring is that the half-life of cell-free DNA is very short. So it's cleared very rapidly within minutes to hours. And that gives us a really nice insight into what's going on in the patient at that particular moment. As already highlighted, I have said it's very short molecules, so we have to think about 50 to 100, 200 base pairs in general, with a clear peak around 166 to 167 base pairs. This is because the DNA is wrapped around the nucleosome and is protected against degradation to a certain extent. If we talk about concentrations, they are varying very much upon whether the patient is in a pathological state, yes or no. So we see in our cancer patients that that dramatically goes up, these cell-free DNA levels. But we also know that after intensive sports, for example, cell-free DNA levels can temporarily peak because of release upon cell death. So the origin, it can be derived from healthy tissues, such as muscle tissue in the case of sports, fetal tissues, tumor cells, but also transplanted organs and microbial. And in the case of this presentation, I will mostly focus on the microbial sources, which you have to be aware of. It's a very small fraction of the total cell-free DNA pool. So try to kind of visualize this here on the left bottom, where the microbial cell-free DNA is less than actually the mitochondrial DNA fraction. And the microbial cell-free DNA consists largely of bacterial cell-free DNA. And sometimes we find molecules that are of viral or fungal origin. So we're talking here about really needles in a haystack that we try to identify for pathogen detection in the clinical setting. So as mentioned already, cell-free DNA analysis, we think it's a very suitable tool for minimally invasive monitoring, a real-time insight into disease status, and supporting early detection monitoring, as well as personalized treatment. And that was also the reason to actually start working in the first place on pathogen detection within Utrecht by the use of liquid biopsies. If we talk about, is there actually a need for better, faster, and more reliable diagnostics within the medical microbiological field? Yes, there is. A large part of the field still depends on slow techniques such as culture. And actually in the critical patient care, there is an essential need for faster and more timely initiation of effective therapy. And therefore you have to know which pathogen is actually positive. This is especially critical, of course, in vulnerable patients that already are in an unhealthy state and which can, suffer much quicker from critical symptoms. And the high quality diagnostics in the medical field, they are sometimes available, but definitely not always. So we, in collaboration with the Princess Máxima, started to see where we can actually use our tool, namely next generation sequencing, to add to the toolbox that is already available. And this toolbox that is already available is mostly depending, as I mentioned already, on culture for bacterial detection, which is very slow. It takes days to weeks. And also the sensitivity and the specificity are relatively low and moderate. Microscopy is still used, of course, the PCR, which is fast but very targeted. And we have the ELISAs that are often used. They're fast, but they have moderate sensitivity and sometimes even low specificity. And with the next generation sequencing, we think we could be relatively fast. We hope to have a high sensitivity, and I will also show you some of these results. And especially the specificity is very, very high, also based on prior research. So with that in mind, okay, we have this opportunity with cell-free DNA shotgun next generation sequencing, but where is there actually a high need for better diagnostics? So as I told you already, we thought that it might be most interesting in a very vulnerable population with a weakened or still developing immune system that are at risk for severe infections. So in this pilot research that we initiated, we started working on an immunocompromised patient population, children under the 18 years at risk for or having a pulmonary invasive mold disease. So this is actually a fungal infection that occurs in the lungs, and these fungi can enter simply by breathing because they're all around us. Early detection here is very critical because these patients are extremely vulnerable, and what is mostly interesting is that currently diagnostics are not really good at pathogen identification at the species level. Well, this is really interesting for this patient population because it does affect which treatment you want to give. So not all species you want to treat with the same antifungal treatment. So we thought it's good to investigate if we can do this in a rapid sensitive and species level dependent manner. So, as mentioned, we thought invasive mold disease would be of interest, and this is also because it's a major health threat in the immunocompromised patient setting. So the incidence is between 15 and 20% in the Netherlands, despite already giving antifungal prophylaxis to most of these patients. The mortality rates are still substantial, and we see about 20% to patients here in the Netherlands per year. So it's a small group, but with a high mortality rate, where we think we can really make an improvement by having better testing methods for microbiology. So the risk factors is the immunocompromised, either with neutropenia, leukemia, bone marrow transplants, or corticosteroids treatment. And actually, in general, there is a kind of diagnostic challenge within this patient group. So they often have non-specific symptoms, limited antifungal options. And what's also important to mention is that most of the antifungals that are given have actually toxicity. So if I talk about non-specific symptoms within this pediatric invasive mold disease population, and then specifically when we focus on pulmonary invasive mold infections, what we see, these patients generally have a fever, maybe they have a non-productive cough and some pain in the chest, but it's very, very vague. So we largely depend on other methods to either see if there's actually something ongoing in the lungs and what's the causative agent. What we know from the literature is that in the majority of the cases, actually an Aspergillus fumigatus that is causative. So in about 53% of our cases in the Máxima Center, it's caused by Aspergillus fumigatus, but there's also other Aspergillus species that can be causative, as well as, for example, the Mucorales species that are also mentioned here in this slide. So how to go about diagnostic currently in Europe? So what we do is we have a classification scheme, which is mostly used for the research setting, actually. So if you have a positive mycology or a histopathological observation in a sterile sample obtained in a sterile manner, then you can actually have a proven invasive aspergillosis. And otherwise, you depend mostly on the host criteria, clinical criteria, and the mycological criteria to actually be able to either classify as a probable or as a possible IMD. If we talk about these mycological criteria, they mostly depend on microscopy, culture, as well as PCR and the galactomannan test. The galactomannan test is actually an ELISA-based test where you look at markers for the cell wall. What is important here to mention is that the sensitivity in general is very low, and specifically if we look at the PCR and the galactomannan, is what we see is that sensitivity is affected by antifungal treatment. And of course, most of our patients are under antifungal prophylaxis. So in the case of, for example, the BAL PCR, we see a sensitivity of less than 1%, which is extremely, extremely low. For the BAL, we see a sensitivity of about 60%. However, we have to mention here that the specificity of the galactomannan is limited. So still, there's lots of diagnostic uncertainty within this population. Now, as a consequence, 50 to 70% remains classified as possible, where we don't really know if there is an invasive mold disease, and if so, which species is actually causative. So we keep them under prophylaxis. We don't really know how to treat. Do we treat them right, or is there under-treatment? At the same time, we have a population where, for example, the galactomannan test is positive, the PCR remained negative, and we don't really know what is causative, which species is present, and if there's actually an Aspergillus present. So we continue with high antifungal doses, not sure if this is actually needed. So in short, there's many reasons to kind of come to the conclusion that the existing diagnostic tools for pulmonary IMD remain inadequate. And this is especially a problem at the early phase of the infection because we saw low sensitivity and specificity. So then we took a dive into the literature and we started looking into, okay, cell-free DNA shotgun NGS. We think it has potential in this setting with what is already known. And most of the work is actually executed by Cadius. It's a commercial test that's available and founded in the US. And it's focusing on broad range detection of infections causing agents, bacteria, viruses, fungi, and parasites. And most of the literature is actually focusing on the sepsis setting. In 2016, I think the Blaukamp et al. Paper has been published. It's a clinical validation study where they see good agreement with blood cultures, and they say also that they are faster than with standard diagnostics, which is of course very interesting, right? Because we say, okay, we're looking here at a vulnerable population that we want to treat as well as possible, as quick as possible. If they test the clinical impact of the carriers test in a real world setting, they say that they can see that there's actually a clinical benefit of using this test to be more targeted in the antimicrobial treatment. But of course, this is mostly focusing on bacteria and sepsis. Then looking specifically at the literature on detection of fungal DNA, what we could see is, yes, there is a certain promise in IND diagnosis. However, most of the literature was focused on testing vicarious, so outsourcing it, and we wanted to really test this in-house, as well as most of the literature is actually focusing on adults, and of course we're focusing on a pediatric setting. So some of the general challenges that we identified is a lack of standardization for in-house testing, because it's very uncommon to do. And also what we saw directly in the literature is that there's ultra low abundance of pathogen cell-free DNA, and that's actually limiting sensitivity in general. So we try to tackle these two actually, and maybe before going into the tackling of these two problems, I just wanted to give you an intuition of, how extremely rare are these microbial cell-free DNA molecules. If we look at, for example, the detection of fetal cell-free DNA, such as used in a non-invasive prenatal screening, you think about maybe 10% of the total plasma cell-free DNA being from the fetus. In the case of tumor-derived cell-free DNA, these levels in the adult setting might be between 0.1 and 5 or 10%. In pediatric cases, we see sometimes levels up to 80% of CT, so cell-free DNA derived from the tumor. And then if we focus on microbial cell-free DNA, it's often very low. So we talk about 0.01 or 0.001% of the cell-free DNA that is actually derived from the pathogen that is causative. So we're really looking for a needle in a haystack, and that's, I think, something that is important to keep in the back of your mind. If you develop something like this, you have to be sensitive and you have to be very specific. So it's posing a challenge, a detection challenge. So it's like finding Waldo in this picture. I can tell you Waldo is actually there, but you need a sensitive method to actually detect this and to identify. And also we want to have high specificity, because of course, I already told you, in the IMD setting, we do have the galactomannan, but it's very lowly specific. So we want to have something better than that. And of course, you want to be fast and point of care. So that's a bit about the background. And one of the gaps that I already told you about is that there's not really a good workflow publicly available for cell-free DNA sequencing with the focus of fungal detection. The general workflow for these types of studies, however, is sample collection, after which you do your cell-free DNA isolation, library preparation, and then transition into next-generation sequencing or even third-generation sequencing. The computational analysis, which is very important because it actually per read identifies the taxonomic origin. So whether a cell-free DNA read is derived from the host or from a pathogen. And based on these taxonomic labels and the quantities of reads observed for specific taxi, you can actually do pathogen detection. And this is often done by comparing to a healthy control population. So this is the general workflow for shotgun cell-free DNA sequencing for pathogen detection. And we started to work on optimizing this actually for the purpose of fungal DNA detection in liquid biopsies in our center. What we saw thereby is that it's unclear how to best recover fungal cell-free DNA from a liquid biopsy sample. It's unclear if blood, or for example, a more invasively collected long lavage fluid holds most potential. And as I mentioned already, there's a lack of standardization of the sequencing data analysis, because most of the literature is actually based on Karius's work, which is a bit more of a black box, right? So we try to tackle these three in a proof of principle study with 7 probable invasive pulmonary aspergillosis patients. And actually our work has been published. So if you want to read more, you can find it online. And I will also show some of these results in the presentation here. And then mostly focusing on recovery of fungal cell-free DNA. So the identification of the needle in the haystack. So now I want to go back to a publication of 2016 of Burnham et al., which was actually focusing on the difference between single-strand and double-strand ligation. And what they clearly show is that with single-strand ligation, you can actually enrich for sub-100 base pair fragments. And what is interesting here is that specifically the microbial cell-free DNA fraction seemed to be ultra-short. So it seemed to be sub-100 base pairs. So the conclusion also that they draw in this specific paper is that library preparation by single-strand DNA ligation gave rise to up to about 70% fold increase in the relative genomic coverage of microbial species. Thereby, it's important to mention that this publication specifically, or well, not specifically, but largely focuses on bacterial cell-free DNA. And of course, we didn't know really what our fungal DNA would look like, but we had a hypothesis that maybe also the single-strand DNA ligation would increase the fungal genomic coverage compared to double-strand DNA ligation. Despite that, we also had in the back of our mind that fungal DNA is chromatin bind and might therefore benefit less from a single-strand DNA ligation method. But it's something that we wanted to test within this pilot study. So then looking for easy to use library preparation kits, we came along the publication of Troll et al. In 2019, where they talked about single-strand library preparation method to analyze cell-free DNA and synthetic oligos. And that actually makes use of the SRSLY technology, which of course you're probably familiar with. So what you do is you denature the DNA before a capture of the cell-free DNA with a random overland adapter. And what's the benefit of this? Well, in the case of cell-free DNA, it's very interesting to look at our short fragments because they are mostly our microbial cell-free DNA molecules. We expect them to be short and fragmented. An additional benefit is actually that you can look at the native termini of these cell-free DNA single-strand molecules. So with that in mind, we thought, okay, maybe we should use this SRSLY kit and compare it actually to a double-strand kappa library preparation to see what is best for fungal detection. So we started doing this in our proof of principle study, where we had both EDTA plasma as well as bronchoalveolar lavage fluid available. And we applied both the single-strand and double-strand ligation protocols to these samples and compared the fungal abundance that we actually observed. So just to quickly highlight the difference with a double-strand library preparation such as used by us, kappa, it does end repair, and therefore your end motifs are not completely retained as they were, which is contrary to the SRSLY library preparation. As I mentioned, the idea was to compare the single-strand with the double-strand library preparation, but of course we needed still an open source analysis workflow to actually be able to identify the fraction of fungal cell-free DNA after both library preps. I don't want to go into too much details, but we have been working on developing a pipeline that is freely available on GitHub that aims to reach as high as possible true positive accuracy by minimizing false positive taxonomic mislabeling. And how did we try to achieve this? Actually, in two different manners. We really try to first identify the host-derived reads, which is like 99.9% of the reads that you actually have in your cell-free DNA molecule sequencing. So we really try to first identify all host reads that are actually in your sample, and then the remaining reads we try to classify as good as possible with a reference database that was curated and developed specifically for the detection of Aspergillus in our samples. So how do we go about this? We first do some QC, then we do some host read mapping, and then we use Kraken 2 to do the classification of the non-host reads. So Kraken 2 is a tool that depends on a big reference database. And in our case, a reference database was composed of a compendium of human genomes, bacterial, viral, plasmids, fungal genomic sequences. And all of this was used to build a database that consisted out of short k-mers. And these k-mers are actually matched to your cell-free DNA readout. And based on the k-mers, either yes or no matching your sequencing read, you can determine the origin of your specific read. So in this case, for example, read A was classified as human, read B was classified as Aspergillus, read C, we did know it's kind of a fungal read, but we couldn't really classify it, for example, at the species level. So what Kraken does, it does classify within the taxonomic tree at the read level. So the output is that you get per read a taxonomic label, and this can be at the species level, genera level, or even higher up in the taxonomic tree. So if you want to know more, the code is available on GitHub. In case if anyone is interested, then you can also drop me an email if you have any questions about this. So going back at what our aim was to compare the single strand with the double strand cell-free DNA ligation method, right? Both in the bronchoalveolar lavage as well in the plasma. And after all this taxonomic labeling, we could actually deduce the fungal fractional abundance. And what we could see, and it was actually non-significant, but what we saw as a trend is that we saw in general more fungal abundance in our single-strand library preparated samples compared to the double-strand library preparated samples. So based on this limited set of samples and data, we thought, okay, we expect that actually the SRSLY prep yields a better diagnostic performance, and we decided to continue with that in the rest of our pilot study. This was not the only question, right? We were interested in, okay, how to best prepare your library. We were also interested in looking at what is better, bronchoalveolar lavage or plasma. And that's actually a comparison that we try to highlight here. Again, there's no significant difference between the BAL and the plasma. However, we decided for now to continue with both because, yes, in general, we seem to have higher levels of fungal reads within our single-strand library preparations if we look at the bronchoalveolar lavage samples, but we still seem to detect signal in our plasma samples. And of course, plasma is minimally invasive, you don't need anesthesia, and is more suitable for monitoring. So we think from a patient perspective, it's very interesting to keep on following up on this, and we tried to keep these samples actually within the pilot study. From here, I want to deviate a little bit into the field of fragmentomics because I think it's highly relevant actually as a background for the story. So this is no longer in the paper that I mentioned, but I think it's an interesting thing to think about that, yes, we know a lot about cell-free DNA fragmentomics, especially if we think about plasma, right? There's lots of literature about fragment lengths and motif-jacked ends, etc., etc. And also we know a part of the biology that's behind it, right? That the cell-free DNA is protected against degradation by the histones, by the transcription factors, etc. So it's a relatively well-established field, and there's plenty of literature about DNAs as mediated cell-free DNA, degradation, nucleosome positioning, etc., etc. But one of the key questions that we had is like, okay, but how does host cell-free DNA looks in immunocompromised patients, right? Because we know their immune system is altered. Does this affect also the cell-free DNA fragmentomics that we see in the liquid biopsies? And specifically, what do we see for the fungal cell-free DNA? Because the logic was to use the single-strand DNA library preparation because we expected our fungal cell-free DNA to be more fragmented and thus shorter than the host cell-free DNA. And we wanted to test this hypothesis. So then looking at the length profiles of the host DNA in the different plasma samples, either after kappa or SRSLY library preparation, what we could clearly see is, yes, we see the normal 166 base per single chromosome peak, and yes, we see a clear 10 base per oscillating pattern, specifically in the single-strand library preparation. So our conclusion was, in short, length profiling aligns with the literature, no major cell-free DNA changes despite these patients being immune and compromised and hematopoietically altered. Then diving into the Aspergillus, so the fungal cell-free DNA part, it's important to mention that we have very little reads to work with, right? So it's really the needles in the haystacks that we combined of the different patient samples to look at the length profile. And I think what's mostly interesting in the single cell library preparation setting is that in general, yes, we see that fungal cell-free DNA is shorter than the host cell-free DNA. What is an important note here is that most of our cell-free DNA Aspergillus reads were from patient 1 because we just had a much higher signal here. So it might be slightly biased to the length profiling of Aspergillus cell-free DNA in patient 1 compared to the other patients. Then if we look at the bronchoalveolar lavage fluid, and again compare the host to the Aspergillus, if we start with the host cell-free DNA length profile, we see that in general, the long lavage cell-free DNA is longer than what we observe in the plasma. In particular, we see for the single-strand library preparation that there's really high periodicity. However, it's really hard to rule out what the storage-related conditions are, because we know that the bronchoalveolar lavage after collection wasn't stored at a cold temperature. It was stored at it was kept at room temperature for a certain amount of time and without any fixative. So we don't really know if there's, for example, genomic DNA contamination occurring here. So I don't want to overinterpret these results. However, again, if we compare the host to the Aspergillus cell-free DNA fraction, then yes, we see that fungal cell-free DNA is in general shorter than host cell-free DNA. So our hypothesis seemed to hold true. Then continuing to our proof of principle study. So back to the paper. This paper contained a small cohort of pediatric patients with a probable invasive pulmonary aspergillosis. So that means that we either had a positive PCR or a positive fungal culture for Aspergillus fumigatus in our bronchoalveolar lavage fluid samples. So for these patients, we had plasma and control plasma available, as well as bronchoalveolar lavage, as well as for 2 other patients, we only had long lavage fluid available. So we sequenced these patients, and we also included immunocompromised control patients where there was no evidence of a fungal infection. And we had in total 9 plasma and 9 long lavage samples available to estimate or to get an insight into the background levels of fungal cell-free DNA and specifically Aspergillus cell-free DNA within this population. And what we see then in our proof of principle is that 5 out of 7 long lavage samples tested positive when we compare to the control samples, and 3 out of 5 plasma samples tested positive when we compare to the control samples. So that means in total, 86% of our invasive pulmonary aspergillosis patients showed evidence by cell-free DNA shotgun sequencing after using the SRSLY kit had a positive result in at least one of our liquid biopsies. And we think this is very interesting because we can identify the species level in a relatively short time frame. Of course, there's also results that we're still a little bit puzzled about. One of them is patient 5, which had a negative long lavage and a negative plasma. And then if we look earlier in time, for example, 24 days prior to diagnosis, we actually see that this patient in a plasma sample showed signal of an Aspergillus fumigatus. And we're just not sure what's going on here. What are the timelines? Is this a false positive 24 days prior to diagnosis, or is it actually a true biological signal? And this is actually something that we're still trying to understand. And I think we simply need more patients to actually be able to interpret these results better. However, I wanted to mention that our external control samples, so the 9 immunocompromised control patients for the long lavage and the plasma, if we do a leave one out analysis, none of them tested positive for Aspergillus species in general. So the take home that I wanted to give you for this part 1 of the presentation is that the single-strand cell-free DNA NGS has potential for diagnostic cell-free DNA setting, where we focus specifically on fungal and even more specifically on Aspergillus detection. Our proof of principle showed elevated Aspergillus fumigatus levels in about 70% of the bowel samples and about 60% of the plasma samples when compared to external controls, whereby we see a very high positive predictive value. But as I mentioned, the cohort is very, very small. So I don't want to overinterpret these percentages. So definitely, like, we need a bigger cohort to validate, but also there's some practical considerations here. If we think about, for example, clinical implementation on the long run, if the sensitivity and specificity would turn out to be good enough, and one of them is that the speed limitation is the turnaround time is simply limited by the time that is needed for the sequencing. And the second thing is that costs are still substantial, right? So you're talking here about €2,500 per sample, which is not extreme, but of course it's something that we have to keep into consideration. So we thought maybe the fragmentomics, on which I showed you already a little bit of data, maybe this gives us opportunities to improve the efficiency by leveraging the cell-free DNA fragmentomics to enrich for Aspergillus DNA. So actually, can we make it cheaper, faster to identify the needle in a haystack? So for that, I want to take you back to prior work and the literature. So yes, there has been already publication on fragmentomics-based enrichment, including size selection for viral and human-derived cell-free DNA, which they showed results in an enrichment of certain viral sequences of certain species, up to 17-fold, and the same holds true for bacterial cell-free DNA fractions. If you do a sub-100 base pair enrichment, that you can actually increase the relative abundance of bacterial cell-free DNA in your sample. Then something else, if we look at cell-free DNA fragments and the motive ends, which is another characteristics of cell-free DNA fragmentomics, is that Dennis Lowe et al. Already have a paper on cell-free DNA fragment-derived end motifs, and that they show that you can kind of discriminate between disease-causing microbes versus contaminated microbial DNA based on the observed and expected end motifs. So with this in our mind, we actually decided to look one more time at our fragmentomics characteristics within our small cohort. And I want to show you quickly the results that we see here. So if we focus on the single-strand library preparation and specifically focus on plasma, what we can see is that Aspergillus fumigatus is shorter than the host fraction. And if we then look at the cumulative frequency ratios, we can see that if you enrich for sub-150 or sub-100 base pair fragments, that you kind of boost your Aspergillus fumigatus to human ratio. So that's actually something that in silico we apply to our patient samples. And unfortunately, we saw that if we would in silico apply sub-100 base pair selection, that unfortunately 1 out of the samples that tested positive prior with shotgun cell-free DNA sequencing now was no longer positive, which means that yes, in general, you enrich for Aspergillus fumigatus, but the downside is that you also lose some signal in some of your patients, which results here in a false negative. This was not the case for our limited set of bronchial alveolar lavage fluids. So I think it's something that definitely would be worth investigating, right? By actually applying this in the wet lab to see what a sub-100 base pair size selection could do. We think it could increase the Aspergillus fumigatus cell-free DNA fraction by up to 8-fold. But yeah, there's this risk of missing positive samples. And I think from a clinical perspective, that would of course be very, very sad. So just the first result to keep in the back of our minds, but I think validation is highly needed. And I'm a bit cautious about implementing this strategy. Then the other alternative is end motif profiling to see if we can enrich for Aspergillus cell-free DNA. As mentioned already, the SRSLY kit is able to capture the native termini. So what we actually did is end motif profiling of both the Aspergillus fumigatus as well as the host reads. And an observation that we have is that fragments starting with a CG show a higher Aspergillus to host ratio. And this is likely because in the human genome, CGs are very, very rare and much rarer than actually in the fungal genome. So in silico, we see some potential to boost the Aspergillus detection. But again, I think this is something that we would have to test in the lab to actually see if this is relevant and if this can actually help us to identify the needle in the haystack. And thereby, I want to make an important note that if we look at the end motifs within our cohort, specifically within the single-strand library preparations, is that we see a certain clustering in the end motifs, and then it seems to be that this end motif signal or abundance is slightly patient dependent. So I think that's something that we should keep in the back of our minds. Not every patient is the same. Cell-free DNA fragmentation is ongoing, but not all of these patients are in the same disease state, right? They're immunocompromised. Some might have more immune cell activity, others might have less activity, and it might also result in less or more DNA activity and also end motif abundance, relative abundance variation. So yeah, we see that these end motifs vary across patients, and for further follow-up and larger cohort is definitely needed to better understand if we can use this. But I hope this second part sparked a little bit of interest in thinking about fragmentomics after a single-strand library prep. How can you maybe use this to enrich for your signal? In our case, Aspergillus fumigatus. We see that this could enable us to really reduce the sequencing cost, right, up to 85% in the case of the lung lavage samples without a loss of diagnostic yield. So if we talk about cost per patient, this could have an impact. I also talked about end motif-based selection, which could potentially boost the fungal cell-free DNA fraction. But all of this is still, as I mentioned, in a preliminary state where experimental validation is still required. And I think to really get an insight into if these fragmentomic-based approaches, but also if cell-free DNA, NGS with the current workflow could hold diagnostic value, we really need better cohorts and bigger cohorts. So one of the steps that we're taking within the Princess Máxima at the moment is the Venice study, where we really try to collect plasma samples at different time points to see if we can use it for monitoring. And another study is that we're starting end of this year is to extend our cohort with many, many more control samples and also include non-Aspergillus invasive mold disease cases to see if this test would also hold true for non-Aspergillus cases. And we know that that will require computational fine-tuning, extension of the classification database, and we're investigating how to do this together with the Westerkijk Institute in our campus. So with that, I think I have to acknowledge all the collaborators. So it has been a pleasure to work with the different centers on our campus. I already mentioned the Westerdijk, which has a lot of experience on fungi in general, the UMC Utrecht, the Wilhelmina Kinderziekenhuis, but as well as the Princess Máxima Center and all our collaborators, as well as the funding agencies, which together made it possible to do this proof of principle study. And it will also will be the team to actually do the follow-up research in the next years. So thank you very much for giving me the opportunity to present here today. If you want to see more of the data, here you have the QR code that is actually giving you access to the full PhD thesis, which also contains the fragmentomics analysis. If you have any questions related, you can also just drop me an email with the email address that is shown here. So thank you very much.
Thank you very much, Emmy, for this wonderful talk and showcasing the application of cell-free DNA in the detection of pathogens. I don't see any questions in the Q&A section, but I have two that I can ask you. One is when you were talking about the time course series where you were looking at plasma and the lavage samples trying to detect Aspergillus. The patient number 5 where you did not detect any, was there anything unique about their clinical symptoms that differentiated them from the rest of the group? Or was there any other infection that you detected, or was there any other treatment that they were undergoing that would, result in the negative results that you saw?
Yeah, that's a very good question. We tried to look into that. I'm not a clinician, so we worked together with the clinic and we actually looked into that. There was no clear indication that, yes, there's something very distinct in this patient 05. I think what is important, especially for the lung lavage samples, that the lavage samples that we received were leftover material. And these were obtained from one of the lung lobes, but we didn't really know from which lung lobe they were actually collected. So it could be that we actually were analyzing a lung lavage sample that is coming from a part of the lung that is not affected by the infection. And that could, of course, cause false negatives. So actually in the follow-up study, we tried to really work on this annotation, like from which part of the lung is the lavage sample taken? And if we look at the scans then, was there also signs of infection? Because I think that could have an impact, right, on negative samples that we see now. Maybe they're taken from part of the lung that was actually not infected, if we look actually at the scans. But we don't have this data available. So this is some of the things that we're speculating about, but we don't know actually.
That's really fascinating because we talk about, tissue of origin and where the cell-free DNA or, DNA is coming from. And if, the lobes can show a differential signal, I think that's very fascinating to look into. I do have another sort of technical question. So in your fragmentomic analysis that you were doing, did you modify your extraction method further because you're seeing a lot of really short fragments and the extraction method can affect what molecules enter your library. So based on those findings, did you change any of your methods?
Yeah, that's a very good question. We are aware of indeed that the cell-free DNA isolation kit can, benefit or work against you, depending on which fragment size you're actually interested in. What we decided upfront, based on the literature that was available at the time, is that the circulating nucleic acid kit from QIAGEN would be a relatively good opportunity to use. I think maybe we could, relook at the literature to see if this is actually the best method possible these days. But we haven't tuned anything since. So we made kind of an educated decision up front and decided to stay with that up till today. But I think it's a very interesting point that you're raising, because indeed, this first step of isolating cell-free DNA can already affect very much of what you're isolating, right? And we might miss some signal here already. That is extremely important that we keep as much as possible fungal cell-free DNA in the isolation, and we haven't just looked at it yet.
I think we have a question in Q&A. I'll read it out. Does Aspergillus have standard CpG methylation? If so, have you thought about performing cell-free DNA methyl-seq to see if that increases signal?
Very interesting point. I actually never really thought about that. But it would be definitely interesting to look into. Yeah. It's maybe something that we could discuss with our collaborators in the Westerwijk, because they do also nanopore sequencing, so they probably know exactly how the methylation looks for these fungal species, and if there's variability also between different species. Because of course, for now we've been focusing on Aspergillus fumigatus, but we really want to expand to all clinically relevant species. So I think we should then have a good map of how methylation looks in all clinically relevant species, and try to avoid that we focus too much on the Aspergillus only. But yeah, very interesting question and suggestion.
All right. If there are no more questions, I'd like to thank Emmy again for a wonderful talk. The kind of scope of work is very impressive. So I'm really glad that you were able to share your work with us. To close out, I think I'm going to segue from the question that was asked at last. We do have a new product where we have a version of SRSLY where we provide methylated adapters. And the idea is that you would be able to do both fragmentomics and metallomics from cell-free DNA. We will share additional information and follow-up emails. And if you have any questions, if you want quote requests, please do write to us at info@clarebio.com. And also, if you want to place an order for a new kit as an attendee, we'll be offering you 15% discount on our kits. All that information will be available in the follow-up emails. With that, I'd like to conclude this webinar, and thank you once again, Emmy.
Thank you very much.